Hi, good morning. This is Kavita Kundra, and we are covering next part of carbohydrate metabolism, that is part 2 of MSCDFSM biochemistry. I hope you read the first part nicely and you learned that. It is very important to learn the basic concepts, also the cycles. So energy production and glycolysis. So ATP generated reactions are when glyceraldehyde 3-phosphate gets converted into 1,3-bisphosphoglycerate, 6 ATPs are formed. When 1,3-bisphosphate phosphoglycerate gets converted into 3-phosphoglycerate, 2 ATPs formed. And when phosphoenol pyruvate gets converted into enol pyruvate, 2 ATPs formed. Then there are ATP utilized reaction where ATP getting utilized for these reactions, like glucose gets converted into glucose 6-phosphate by utilizing 1 ATP, and fructose 6-phosphate gets converted into fructose 1,6-bisphosphate by utilizing 1 ATP. Therefore, the net ATP generated is 8. How it is 8? 6 plus 2 plus 2. It's 10 minus 1 and 1 is equal to 8. In anaerobic conditions, the total number of ATP will be only 2 up to lactate. The generation of NAD plus from NADH formed in the reaction of glyceraldehyde phosphate dehydrogenase step is utilized by lactate dehydrogenase to form lactate. 2 ATP molecules are synthesized in anaerobic glycolysis. Regulation of glycolysis. How the glycolysis gets regulated? What regulates that reaction? There are 3 exogenic reactions in glycolytic pathways that are irreversible. These reactions are catalyzed by 3 enzyme hexokinase, phosphofructokinase 1 and pyruvate kinase. These reactions are the major sites of regulations. Phosphofructokinase 1, it is activated by AMP and inhibited by ATP and citrate. When ATP gets utilized in energy requiring process, the AMP concentration increases, and these large quantity of AMP act as amplifier of small change in ATP. Due to this, PFK1 become highly sensitive to even small changes in energy, and to control the quantity of carbohydrate undergoing glycolysis before entering into citric acid cycle. When ATP decreases, the glycolysis increases due to increase in AMP. The regulation of glycolysis by allosteric activation or inhibition or phosphorylation dephosphorylation of rare limiting enzyme is short term. Consumption of a meal rich in carbohydrate or administration of insulin results in the increase of glucokinase, phosphofructokinase 1, pyruvate kinase in liver. These changes result in enzyme synthesis, and these enzymes convert glucose to pyruvate. When plasma insulin is low and glucagon is high, this results in the decrease of enzyme in case of starvation or diabetes. Inhibitors, iodoacetate, it inhibits glyceraldehyde 3-phosphate dehydrogenase. Arsenite, it inhibits the synthesis of ATP in conversion of 1,3-bisphosphoglycerate to phosphoglycerate by uncoupling the oxidation and phosphorylation in mitochondrial ETC electron transport chain. Fluoride, it inhibits enolase enzyme involved in the conversion of 2-phosphoglycerate phosphoenol pyruvate. Oxidation of pyruvate to acetyl coenzyme. The oxidation of pyruvate to acetyl coenzyme is irreversible route from glycolysis to citric acid cycle. The citric acid cycle is cycle that stores energy released by oxidation of carbohydrate, fat and protein Pyruvate must be transported into the mitochondria before it enters in citric acid cycle via special pyruvate transporter. It involves a support mechanism where one proton is co-transported. In this case, pyruvate and hydrogen are transported from cytosol to mitochondria. Oxidative decarboxylation of pyruvate to acetyl coenzyme occur inside the mitochondria, and it is catalyzed by multi-enzyme complex called pyruvate dehydrogenase complex, which is present in mitochondrial matrix. This irreversibility proves that why glucose cannot be formed from acetyl coenzyme in gluconeogenesis. The PDH full form is pyruvate dehydrogenase complex. It is multi-molecular aggregate of three enzymes named pyruvate dehydrogenase, dihydrolipol transacetylase, and dihydrolipol dehydrogenase. Reaction involved in the oxidation of pyruvate to acetyl coenzyme. There are four different enzyme activities. Each enzyme activities require different substrate and cofactor that comprise enzyme complex. First, oxidative decarboxylation of pyruvate. This reaction is catalyzed by EI subunit of PDH. E for E1 subunit of PDH. The cofactor thiamine diphosphate is required. In this step, carbon dioxide is formed and a hydroxyethyl derived from pyruvate covalently bond with TDP. Second, transfer of the 2C unit from E1 to E2. E2 requires lipoic acid. It is a coenzyme that is attached to lysine residue of enzyme protein, and it is called lipoamide. In this step, a hydroxyethyl group is simultaneously oxidized to acetyl group and transferred to oxidized form of lipoamide. TDP is regenerated in this step. Formation of acetyl coenzyme is the third step. This reaction is catalyzed by E2 subunit of PDH. This step involves the transfer of acetyl group from lipoamide to coenzyme A to form acetyl coenzyme. After this transfer, lipoamide is in reduced state. Regeneration of lipoamide. This reaction is catalyzed by E3 subunit of PDH. The cofactor here are NAD plus and FAD. FAD reoxidizes lipoamide and is reduced to FADH2. Now the turn of FAD. FAD regeneration of oxidized FAD. FADH2 is reoxidized to FAD by NAD plus. NADH and H plus are produced. NADH and H plus are oxidized in adjacent electron transport chain, forming NAD plus and 3 molecules of ATP. Regulation of pyruvate dehydrogenase. The activity of PDH complex is regulated by following factor. First factor is product inhibition. Second factor is substrate availability. And third factor is covalent modification. Product inhibition. Both NADH and acetyl coenzyme inhibit pyruvate dehydrogenase. Availability of substrate. For the complex to function, concentration of acceptor molecule, coenzyme A and NAD plus must be adequate. Covalent modification. PDH exists in two forms: active dephosphorylated and inactive phosphorylated. The active form of PDH is phosphorylated by protein kinase, and with the help of ATP and magnesium ion to inactive form of PDH. Acetyl coenzyme and NADH are the activators of, and coenzyme pyruvate NAD are the inhibitors. They inhibit this reaction. The inactive form of PDH is dephosphorylated by phosphoprotein phosphatase to active form with the help of calcium ions. Genetic defect in pyruvate dehydrogenase. A defect in any of the protein subunits of PDH causes inactivity or decrease in activity. The symptoms are lactic acidosis and neurologic disorders. Chronic alcoholic have thiamine deficiency that results in pyruvic acid accumulation. Excessive pyruvate gets converted into lactate and it leads to pyruvic and lactic acidosis. When there is defect is in E1, large doses of thiamines are effective. When the defect is in E2, large doses of lipoic acid is effective. Keto diet is also helpful in lowering pyruvate and lactate, and it is low in carbohydrate and in high in citric acid cycle, or it is also called Krebs cycle, and another name is tricarboxylic acid cycle, TCA. It is a series of reactions that form a common pathway for final oxidation of all metabolic fuel that are catabolized to substrate acetyl coenzyme of citric acid cycle. Its central function is oxidation of acetyl coenzyme to carbon dioxide and water. This oxidation is a major site for oxygen consumption and ATP production in most of the animals, including human. The enzyme of this cycle is located in mitochondrial matrix in close proximity to enzyme of respiratory chain, thereby facilitating the form reduced coenzyme to oxygen. It is aerobic process. In the citric acid cycle, oxaloacetate is condensed with acetyl coenzyme and then regenerated for oxidation of large quantity of acetyl coenzyme. Small quantity of oxaloacetate is needed. Functions of citric acid cycle. It is amphibolic pathway. That means it involves both anabolic and catabolic pathway. Anabolic reactions are the intermediates of citric acid cycles are used as precursor in biosynthesis of many compounds like synthesis of glucose from carbon skeleton of amino acids and providing building blocks for heme synthesis. Catabolic reaction, this cycle provides the mean of degradation of two carbon acetyl residues that are derived from carbohydrate, fats and amino acids. This cycle generates OTP. This cycle generates ATP by oxidative phosphorylation when the electron generated in the cycle are transferred to ETC and the cycle stores energy. Reaction of citric acid cycle. Synthesis of citrate from acetyl coenzyme and oxaloacetate. Citrate synthetase catalyze this aldol condensation with the release of CoA. Acetyl coenzyme. Inhibitor citrate synthase is inhibited by ATP, NADH, succinyl coenzyme, acyl coenzyme, derivative of fatty acid. This is the citric acid cycle. It is very easy and you please go through it. Pyruvate converted into acetyl coenzyme and it converts to CoA, that is that activates citrate, then it's converted into cis econitate, diisocitrate, alpha ketoglutarate, and succinyl CoA, succinate, then fumarate, then maltate, and oxaloacetate, and again it becomes citrate. It's very easy, it's not very difficult, so please go through it and understand it properly. The role of reaction is also determined by the availability of the substrate. Role of citrate. Citrate is intermediate of citric acid cycle and is also a source of acetyl coenzyme for cytosolic synthesis of fatty acid. It inhibits phosphofructokinase 1 and the rate limiting enzyme of glycolysis. Citrate activate acetyl coenzyme carboxylase. Isomerization of citrate. Citrate isomerized to isocitrate with the help of aconitase, which has iron sulfur center as its prosthetic group. Oxidation and decarboxylation of isocitrate to alpha ketoglutarate. Isocitrate dehydrogenase catalyze this reaction, yielding first 3 NADH molecule and release carbon dioxide. The aim of cycle is to oxidize acetyl units to 2 molecule of carbon dioxide. The enzyme isocitrate dehydrogenase is activated by ADP and inhibited by ATP and NADH. Oxidative decarboxylation of alpha ketoglutarate to succinyl coenzyme. This reaction is catalyzed by ketoglutarate dehydrogenase complex and mechanism of action is similar to PDH. The reaction releases the second carbon dioxide and produces second NADH of cycle. The coenzyme required was same as PDH complex that include thiamine diphosphate, lipoic acid, FAD, NAD+, etc. The alpha ketoglutarate dehydrogenase complex is inhibited by ATP, GTP, NADH and succinyl coenzyme but not regulated by phosphorylation or dephosphorylation. Cleavage of succinyl coenzyme to succinate. Succinate thiokinase cleaves the high energy thioester linkage of succinyl coenzyme and it releases succinate and coenzyme along with the substrate level phosphorylation of GDP to GDP. Succinyl coenzyme is used in biosynthesis of heme. Oxidation of succinate to fumarate. The reaction is catalyzed by succinate dehydrogenase and FAD is needed as cofactor. Malonate inhibits succinate. It is malonate, it's not malonate. Malonate inhibits succinate dehydrogenase and it is completely inhibited by oxaloacetate. Hydration of fumarate to L-malate. Fumarate catalyze this reaction. Oxidation of malate to oxaloacetate. Malate oxidized to oxaloacetate by malate dehydrogenase and NAD+. It is required as coenzyme, needed as cofactor. This is the third step of NADH production in citric acid cycle by electron transport chain along with generation of ATP molecule. Stoichiometry of citric acid cycle. Acetyl coenzyme plus three NAD plus FAD plus ADP plus pi plus 2NH2O get converted into 2 carbon dioxide, 3 NADH, FADH2, ATP, 3 hydrogen ion and acetyl coenzyme. So we can see the summary. What happens is here 2 carbon atom enter the cycle as acetyl coenzyme and leave in the form of carbon dioxide. 4 pair of electrons are released. 3 pair are released in the form of NADH and 1 pair release in the form of FADH2. One high energy phosphate bond is generated in the form of ATP by substrate level phosphorylation. Net energy output of the cycle: oxidation of one NADH by ETC leads to the formation of 3 ATP, whereas oxidation of FADH2 yield 2 ATP. Here, isocitrate gets converted into alpha ketoglutarate, where NADH converts it to NAD plus and 3 ATP gets released. Alpha ketoglutarate gets converted into succinyl coenzyme, and here NADH gets converted to NAD plus, again 3 ATP get released. Succinyl coenzyme gets converted into succinate, here ADP converts into ATP and 1 ATP gets released. Then succinate gets converted into fumarate, where FADH2 gets converted into FAD and 2 ATP get released. Maltate gets converted into oxaloacetate and NADH gets converted into NAD and 3 ATP are released. ATP generated, total 12 molecules of ATP are generated in this reaction. Thank you so much. I hope you enjoyed the session and I hope you will go to the basics. Please go to the basics and start learning, and I hope you can do better in the examination. And any questions you can, if you have, you can ask in the comments, and please share it with your friends so that they can also get the material for studying and get help. If they or you need help of any kind, you can always comment. I will be there to help you. Bye bye, take care, and we are soon coming up with the next section of carbohydrate metabolism soon. Bye bye, stay safe, stay protected. Take care. Thank you.